So welcome to lecture number 17, where we continue on our journey through the gastrointestinal tract. And we're starting on plate 75. We finished with talking about the hormonal regulation of digestion, and now we're going to talk about neural regulation of digestion. And in the upper right hand of plate 75, we revisit once again the sympathetic and parasympathetic nervous system. Remember that the arms of these are divided anatomically, where they arise out of the spinal column, the neurotransmitters, and hence the function. The parasympathetic system is primarily the stimulating nervous system for gastric digestion. This is a vegetative state. You don't need it when it's flight or fright. So the parasympathetic system ramps up activity of the stomachs, of the livers, and the GI tract, increasing motility. In contrast, the sympathetic system tones it down. So these dashed lines indicate an inhibitory response. This is part of the autonomic nervous system. This occurs without your control. We certainly have higher centers involved in the regulation of digestion. And in fact, it's termed the cephalic phase. This, in fact, can occur before food even enters into the mouth. There is sensory input, there's visual, there's olfactory. In fact, there can even be auditory input, sounds associated with feeding. The classic Pavlovian dog, ring the bell, produced salivary secretions. There are then arms of the central nervous system going and innervating directly the glands of salivation. The process is started. You're preparing to take that bolus of food in. Saliva starts being produced even before it enters into the body. One of the major cranial nerves that you learn about in lab is the vagus nerve. The vagus nerve is cranial nerve number 10, and it is one of the major parasympathetic nerves that will start the gastric juice secretion. This all occurs in the cephalic phase. So even before food starts moving down, you start increasing HCl production in the stomach. So in anticipation. Now the majority of acid and the majority of the digestion occurs later in the gastric phase, but during the cephalic phase, these processes are already started. So increase in gastric juices, and you will also see an increase in the pancreatic juices. From the cephalic phase, food then is passed through the esophagus down into the stomach. It is that bolus of food that's entering into the stomach that stimulates stretch receptors and initiates part of the gastric phase. Chemoreceptors, stretch receptors, both sense this food entering into your stomach and further increases gastric juices and increases the muscular action of the stomach. So when you say, oh, my stomach is churning, that's exactly what's happening. You have entered into a gastric phase, and there is in your notes what's called the intrinsic nervous system of the gut wall. So you have both the extrinsic, which are these nerves, like the vagus coming down, the sympathetic and parasympathetic nerves coming down. In addition, there is a neural network that is lining the walls of the stomach. And down here you see these mechanical receptors and chemoreceptors that are responding. These are embedded in the mucosa. So this is where the food is. This is the mucosa. And the submucosa and the muscles that are underlying it. Embedded in that underlying muscle are nerves that are lumped together in what is called the myenteric nervous system. It is these nerves then that can respond directly to these chemical and mechanical stimuli to produce contractions both of the stomach and then later on in the intestine. So this churning is initiated both by the parasympathetic system. The parasympathetic system stimulates it. The sympathetic system inhibits it. But then you have this own myenteric, what's called plexus, or network of nerves. In fact, a number of smooth muscle studies are done in excised GI tract, because you can take it out, and it has both muscle and nerve input. The final phase of digestion is the intestinal phase. Gastric and secretions are significantly lower. This is primarily an absorptive phase, and so these secretions are decreased. But this same myenteric plexus helps stimulate and cause contraction and move food down the GI tract. The pancreas we've mentioned once before, and now it's time to revisit it in a little more detail. If you haven't picked up on it, one of the nice things about this book is it tends to interdigitate or return to many of the topics in different formats, in slightly different ways, to re-emphasize. So the pancreas is a relatively large gland, although it looks pretty fragile, pretty friable. It lies next to the stomach and immediately next to the duodenum. It is both an exocrine and an endocrine. The exocrine role is by and far the largest role it takes, at least in terms of the number of cells. That's not to denigrate or downplay the importance of the endocrine. But in terms of the number of cells that are allocated to a function, it is the exocrine pancreas that constitutes the majority of the cells. Here's an example or a cartoon of these exocrine cells connected together, emptying their products into a tube or duct. This is the pancreatic duct, which exits immediately after the pylorus. So this is the stomach up here. This is the pyloric sphincter, so there's a ring of smooth muscle that regulates it. One of the first things that chyme sees when it comes into the stomach is this pancreatic duct. Recall that very acid, pHs of 1 to 3 in the stomach, must be neutralized to prevent damage to the GI tract. Pancreatic juices, rich in bicarbonate, drop into the duodenum through the pancreatic duct. So these ductile cells are producing large amounts of bicarbonate. You will recall the enzyme that is found in high concentrations in this cell is carbonic anhydrase. We see numbers N hydrase. We see a number of examples of cells whose responsibility it is to either produce bicarbonate, or in the case of gastric cells, to produce hydrochloric acid, a byproduct of which is bicarbonate that's put into the blood. And they all utilize this enzymatic mechanism. So these ductile cells, high concentrations of carbonic anhydrase, they produce the bicarbonate, and it is sodium bicarbonate then that is dumped into the duodenum. In addition to ductile cells, there are what are called acinar cells. Acinar cells, also part of the exocrine function of the pancreas, have as their role the production of a precursor to a protein enzyme. Remember, in the stomach there was pepsinogen. Inactive form, because you didn't want to auto-digest the stomach, you dumped pepsinogen into the stomach. Hydrochloric acid cleaved it to trypsin, it became active. And then the mucus helped protect. A slightly different strategy used by the acinar cells. They produce trypsinogen. Trypsinogen is the inactive precursor. It moves into the duodenum, and along the walls of the duodenum is an enzyme called enterokinase. So anytime you see this prefix, entero, or enteric, it refers to the intestinal tract. So an enterokinase is an enzyme in the GI tract whose responsibility is to cleave trypsinogen to trypsin. These are active digestive enzymes. In addition to production of a protein-degrading hormone, we also see production by the exocrine pancreas cells of a pancreatic amylase, a pancreatic lipase, and other pancreatic proteases. So in addition to trypsin, there's other proteases. Lipase has the responsibility of breaking down lipids. The major storage form of lipids are triglycerides. Triglycerides are broken down into free fatty acids and the glycerol moiety. So you see this kind of representation of this chewing process. So this little enzyme is breaking down triglycerides, making them into smaller, digestible compounds. Amylase was an enzyme that we encountered in the oral cavity. It was being produced by the salivary glands. Well, now we see another amylase, another carbohydrate enzyme, which is being produced by the pancreatic cells. So we have all the components now of digestion. So consider an animal with pancreatitis. Inflamed pancreas. It loses its function for amylase production, lipase, proteases. Now there's going to be some digestion that may occur in the mouth, some digestion that may occur in the stomach, but really the bulk of the process is right here at the pancreas. And dogs with pancreatitis have incredible problems. First of all, it's very painful. If these animals recover and they have inadequate pancreatic function, clinically they present as undernourished. They can eat and they take material, but they aren't producing adequate amounts of amylase or lipase or the proteases to digest it. Hence, they are not absorbing the nutrients. So these animals are voracious. They eat and eat and eat. We refer to that as cachexia. So anorexia, I'm not eating, I don't want to eat. Cachexia is, boy, I want to eat, I am eating, but I'm not gaining any weight. That's the best kind of recovery from pancreas. Pancreatic cancers kill you fast. A pancreatitis untreated or unresolved can kill you. A pancreatitis that resolves has the potential for this cachexic condition. Actually relatively treatable because we now have oral supplements that include amylases and lipases. These come down, are digested, partially digested in the stomach, but enough of them make them into the duodenum so that as an adjunct or a supplement, these enzymes taken orally can help to counter the fact that the exocrine function of the pancreas is reduced. Stimulation of these of this is in part under cholecystokinin. We talked about that in its hormonal regulation. It was important both in bile but also in pancreatic expression. We are going to spend time on the endocrine function in more detail, but this is a great place to start. This is your little block of pancreas, and there's a small little component consisting a cluster of cells. Only 2% of the total population of pancreatic cells are composed of these little islets or little clumps of cells. Named after the anatomist islets of Langerhans, the early anatomists could, by staining techniques, identify 3 different cell types. They called them alpha, beta, and gamma. They didn't know their function. Well, we now know that these islets of Langerhans have an endocrine function. Alpha cells produce a hormone called glucagon, and beta cells produce insulin. Insulin functions in decreasing the concentrations of glucose in the blood. Glucagon increases it. So in addition to the exocrine function, its involvement in the production of amylases, in the digestion of carbohydrates, the endocrine function of the pancreas plays important roles in the regulation of blood sugars. In addition, the gamma cells produce another hormone called somatostatin, which, in fact, can either decrease or increase glucose. It has a multifaceted regulatory component that we'll talk about when we talk about endocrine cells. Okay. Pancreas, major digestive organ that feeds its material in through the pancreatic duct. Oh, sphincter of Odad. So there's a great one. Yeah. And that's in plate 77. So if we turn to plate 77, we're going to revisit some of this in a little more detail. Here's the stomach, first portion of the intestine, the duodenum. There's the pancreatic duct. And you see that at the very end of it, there is also a duct feeding in from the gallbladder. Not all animals have gallbladders, but they all have bile ducts and pancreatic ducts. Largest organ in the body, lobes of the liver. I was listening to a commercial the other day, and they said the major filtrating organ of the body is the liver. Not true. Major metabolic processor of the body is the liver, but most of the filtration occurs at the kidneys, simply by volume of material that's circulated through there. That's not to downplay the role of the liver. The liver is presented with blood and is important in a lot of detoxification. So in that sense, yes, it is acting as a filter, but also major site for protein metabolism. Has a major role in digestion. One of the neat things about the liver is its blood supply. There is blood that is delivered, oxygenated blood that is delivered to the liver by a hepatic artery. So whenever you hear the word hepatic, that makes reference to the liver. And in this diagram, it's showing this blood is coming into the hepatic artery but what it does is dumps into these large spaces. Now, these aren't exocrine functions, so these aren't called duct. Rather, these are called sinusoids, or sinuses. So there's this large pooling of blood. So all of these hepatocytes, or liver cells, are bathed then in blood. In addition, you see blood being delivered from the portal vein. A portal vein is a vein that is between two capillary beds. In the kidney, we saw two capillary beds separated by an arteriole. So we had an arteriole that came in and delivered blood into the glomerular capillaries, and then another artery that delivered blood into the capillaries of the peritubular. And so there was an afferent and an efferent arteriole between two capillary beds. Now what we have is we have a portal vein, so we have an artery coming in, a capillary bed, we have a hepatic portal vein, and another set of capillaries. This efferent arteriole that was found in the kidney is high pressure. The portal vein is low pressure. It's bringing blood from the walls of the intestine. So figure that nutrients are absorbed into the capillaries in the intestine. They are carried out of the capillaries by the flow to a hepatic portal vein. And then they are dumped into these capillary beds, or the sinuses, right here. That is, these hepatocells then are bathed and exposed to high concentrations of nutrients directly from the stomach. Directly from the intestines. In addition, there's this duct. Now, these are sinusoids, so these are carrying blood. There is a duct, so this is not carrying blood. It is rather carrying products secreted by the hepatocytes into this duct. This is the bile duct. This cell represents one of those cells that is involved in the production of what are termed bile salts. In your book, bile salts, also called bile acids, cholic acid and D-cholic acid. Bile salts are critically important in the digestion of lipids. Oil and water doesn't mix. Lipids in aqueous environments are difficult to digest. We talked about the presence of lipase. Lipase is an enzyme which breaks down triglycerides, but one of the problems is enzymes are proteins. They're solubilized. They need to be in an aqueous environment, and they are faced with a lipid environment. So cartoon-wise, here's this little lipase molecule seeing this big glob of fat. Not a lot of surface area. What bile salts do is they emulsify. That is, they tend to break up the lipid droplets. Same thing that you do when you add soap into a fatty mixture. You're doing dishes, it's really lipid-laden, and you need to break up that lipid. You can add in soaps. Those soaps emulsify or help break up those fats. That is the function of bile salts. Here, this bile salt is breaking up this lipid into smaller particles. The smaller particles of triglycerides then can be acted upon by the lipase and converted into these different compounds. This book goes. Part of the philosophy of this book is that if you'd been coloring, you would recognize these little dots as free fatty acids, and this is the glycerol moiety. If you needed to be reminded of that, you could trace back to these little subscripts. So, hepatocytes, bile ducts, bile salts, bile pigments. Bile pigments are pigments, they're colored compounds that arise primarily from the breakdown of hemoglobin. So in addition to filtering material that's coming and processing material that's coming from the intestines, liver is the site of hemoglobin degradation. Where do old red blood cells go? They get broken down. The hemoglobin gets broken down. Hemoglobin breakdown compounds include bilirubin and a compound called urobilinogen. Bilirubin glucuronide. Bilirubin glucuronide. It's this compound that is greenish yellow that gives a characteristic hue to the bile. Bile duct is dropping these bile salts in that helped in the emulsification. They are also drawing into the bile pigments into the duodenum. Well, that's going into the intestines, and that is one of the components that provides coloration for fecal material. So down at the bottom we see red cell breakdown. Breakdown products are picked up by the liver, processed, and then dumped into the GI tract. Gives feces its color. There is some reabsorption of these colored pigments, these bile pigments, represented by this being taken back up into the blood and being filtered. So in fact, part of the coloration of your urine is due to these bilirubin breakdown products. So bile pigments give bile its characteristic hue. It gives the feces and the urine its coloration. Problems with the bile duct? Problems with hepatic function? If you're not able to get rid of those bile pigments, you will have then an accumulation in the blood. That produces a yellow coloration called jaundice. So jaundice, yellow coloration in domestic animals and heavily darkly pigmented animals, you can perceive it in the eyes. The eyes turn yellow. Sign of liver failure, sign of a bile blockage. Both problematic, both need to be treated. Young babies, a not uncommon problem with young babies is an inadequate hepatic function. So as they're still developing, they may show up with jaundice. Another cause of jaundice is excessive breakdown of red cells. If there is so much red cell breakdown that you present the liver with too much hemoglobin, it just can't handle it and process it and get rid of it. So in fact, you see a reabsorption in the blood, and jaundice can occur from excessive red cell breakdown. Control of bile secretion under the control of cholecystokinin. It's named cholecystokinin. Cholic acids, bile. So when you hear the term cholic or chole, that is in reference to bile. Cysto meaning the bladder or the gallbladder, and a kinin to produce movement. So this contraction of the gallbladder is under cystokinin. It is also under parasympathetic control. So this is kind of that duality. You have both hormonal input and neural input in digestion. Turn to plate 78. Talk about the small intestine. In our domestic animals, huge variations in the lengths of the small intestine, which probably has due in large part to what they're eating and the requirements for digestion. If you eat highly digestible substrates, you need a relatively short intestine. More problematic, tougher to digest things like herbs, longer GI tracts. In fact, you can sacrifice a substantial portion of your intestine and still be really quite functional. Chemical digestion starts in the duodenum. It continues in part throughout the intestine, certainly in the duodenum, and also at the same time you have absorption. So the small intestine is broken into three sections. You don't need to know the percentages, but you do need to know the order, from duodenum to jejunum to ileum. At the junction of the ileum, you then move into the large intestines. In a number of our domestic species, there may be an enlargement or pocket of the GI tract sitting right between the ileum and the colon. That is the cecum. In some animals, this is a fermenting vat, helpful in terms of providing nutrients to the animal from difficult to digestive food stuffs. It's also a great place for horses to get colic. If we look at the wall of the intestine in microscopic detail, so this is a slice through it. This is the lumen of the intestine, so food material is traveling in this direction. It's composed of a mucosa, submucosa. It has that myenteric plexus, so it has those smooth muscles, important in the peristalsis, the movement of the food and helping in churning it. It's also intestinal of intestinal folds. Intestinal folds act to, intestinal folds act to increase the surface area. And there's some numbers here. Again, you don't have to know these exact numbers, but have some kind of concept for the magnitude. It's gotten very dark in my life. There's an animal through a snowstorm. Oh, you're looking quite white today, Wickler. Oh, I'm back. It's anemia. But I don't have a pointer. Did I lose my pointer? So get one or the other. Oh, we're fine-tuning it. Well, let me finish this one section. And I can do it with this magic pointer right here. This is my electronic pointer. So it says 3 times, but the whole principle of this involution or folding is to increase the surface area. That is increasing the surface area for absorption. So at the first level, these are what we call intestinal folds. If we take these intestinal folds and we enlarge them, we'll see that each fold is comprised of a number of projections. In the kidney, we talked about a brush border having these little fingers to increase the surface area for reabsorption and secretion. Well, similar in the gut. And you see that there is an increase of what are called these villi. All right, so each of these villi, and these villi are composed of microvilli. So here's a villi. So you'll note that each of the walls is composed of little tiny cells. So these are intestinal cells. On the intestinal cells, there is a further increase in the rugosity, or folds. Those are microvilli. So we go from intestinal folds, which increase the surface area by 10 times, to the villi, which is about 10, to the microvilli, which increase it another 20 times. So if you want to look at how this folding impacts surface area, you can just take the multiplication. You can go 3 times 10 times 20. So, and again, huge amount of surface area. If we look at the individual cells, this is where absorption is going to occur. And we'll talk about that in the next plate. But what we can do now is look more at the relationship of these cells to blood supply. And here, so if we look right in here, am I functional now? Very good. Thank you. So here are these villi, and into each of these projections, so into each of these little fingers, extends a capillary system. So these then are the intestinal capillaries. Material will be absorbed into these and then picked up by the capillaries, transported by the vein to the hepatic portal vein. So a lot of absorption is occurring here. Specifically for fat absorption, however, the fats don't tend to be taken up by the capillaries. The fats tend to be taken up by special lymphatics. We talked about lymphatics being important in terms of recycling fluid, recycling protein, and we also had mentioned fat absorption. That is exactly what happens here, is these specialized lymphatic limbs that move up into the villi are called lacteals. So lacteals are specialized lymph ducts in the intestine which pick up fats and then transport it by a lymphatic system back into the venous system, the same place that it deposits extra fluid from the vascular and interstitial spaces. So all lymphatics converge on a common point in the thoracic cavity called the thoracic duct. All right. I think we'll stop there.